Good morning, good evening. So now we are getting into the day 2, section 3. This would be the last section of this 2-day seminar. We have 3 wonderful speakers for this section titled Emerging Methods for Installing and Manipulating RNA Modifications. The first speaker would be Dr. Paresh Mali from University of California, San Diego Health Sciences. He will talk about the programmable modulation of RNA editing, new approaches and new challenges. So whenever you're ready.
Yeah, thank you, Shenglong, and thank you to Sami and Kate for this wonderful opportunity to present some of our work. Can you see my slides here?
Yes.
Okay, great. So, yeah, I'm going to talk about 2 studies from my lab in this talk. The first is a recently published study, and a second is a still ongoing unpublished body of work. And so I really welcome feedback and comments as we are sort of developing that work. And basically these two studies that I'll talk about also sort of touch upon the 2 major focus interests of my lab. The first is we're very interested in genome and transcriptome engineering, tech dev, but we are very interested in doing this primarily in the context of being able to apply these to study early developmental processes. So the 2 projects I'll talk about essentially touch upon RNA editing, but one in the context of technology development and the other in the context of studying early development. So focusing first on the work on programmable RNA editing. And so in this context, as we think about nucleic acid targeting, one can do this either at the DNA level or at the RNA level. Of course, being able to do it at the DNA level comes in with the advantages that the corresponding changes can be durable and permanent, and they can basically enable a direct therapeutic cure. But being able to also target at the RNA level from a therapeutics perspective opens the door to the fact that you can be tunable and reversible, and also no off targets are basically permanent. And especially in the context of being able to target at each of these 2 levels, especially when you think about RNA, what the past 20 to 30 years of ASO work has shown us is that classically RNA is accessible to short antisense oligonucleotides, either because it is single-stranded in regions, or it becomes transiently single-stranded, for instance, say through the passage of the ribosome. So classically, short antisense oligonucleotides, without requiring any other heavy machinery, can access RNA. And what these can basically do, either by themselves or upon complexation with the target, is they can recruit machinery such as the RISC and RNase-H complex to enable targeted RNA knockdown. But one other very conceptually interesting aspect about this targeting modality is that in these approaches, what is being delivered exogenously into the cell is just an antisense oligonucleotide, but all the remaining machinery to enable targeted RNA knockdown is already present inside the cell. So we were sort of really inspired by this sort of thought, and we started to wonder if we could also extend RNA targeting from the mode of RNA knockdown to also enable RNA editing. And in this regard, we basically relied on a family of proteins called ADARs, and essentially there are 3 major family members, ADAR 1, 2, and 3, as we're all very familiar with. And among these, ADAR 1 is perhaps the most important family member in the sense that it is very robustly expressed in every single cell type in the body. And what fundamentally these proteins can do is they can catalyze the conversion of an adenosine on an RNA into an inosine. And this inosine basically gets recognized by the cellular machinery as a guanine. So for all practical purposes, it's an A to G change. But of course, if you can programmably introduce this A to G change, one may think that, well, this is only one modification, but there's about 25% of human diseases that are caused by point mutations that could potentially be reversed if one could make an A to G change. Additionally, 10% of human diseases are caused by nonsense mutations, be it the Amber, Ocle, or Oper nonsense mutations, and these could also, in principle, be reversed if one could make a programmable A to G change. And so we started to think about considering the use of ADARs, but actually the idea of using ADARs to enable this targeted RNA editing is actually an old one, and it really comes from this classic paper by Wolf and colleagues going back to 1995. And so we were really inspired by this study, and a very talented graduate student in my lab, Dhruva Katrekar, basically set out to build a platform to utilize ADARs to enable targeted RNA editing. And so just as a quick recap of some of Dhruva's earlier work, to just sort of contextualize the more recent study, what basically Dhruva discovered is, as I had mentioned earlier on, RNA is, of course, accessible to short antisense nucleotides. But what Dhruva discovered through a lot of trial and error is if you just simply increase the length of this antisense RNA, then one could recruit ADARs to a target RNA site. And of course, on hindsight, this made sense because fundamentally what ADARs are is they're double-stranded RNA binding proteins. And by extending the length of this antisense RNA, essentially we've created this extended duplex structure that then becomes a favorable substrate for the ADARs to come and dock themselves at. And very interestingly, what Dhruva observed in these studies was that one could, using this long antisense RNA, recruit endogenous ADARs. And especially the exciting aspect about this is ADAR1 is expressed in every single cell type. And so this was now very similar to the RNAi/ASO modalities, where we only need to deliver this long antisense RNA, and then the machinery to do the targeted RNA editing is already present inside the cell. But of course, one can also deliver exogenous ADARs. And again, this simple format of increasing the length of the antisense RNA suffices to enable targeted RNA editing. But in these studies, we also observed sort of two major challenges. The first of these was that while we could recruit endogenous ADARs, our efficiencies of RNA editing were quite low. We were only getting about 10% or so targeted RNA editing at a particular site of interest. Now, we could improve this targeted RNA editing by instead now providing exogenous ADARs. What I mean by that is by delivering ADARs at supraphysiological levels, by, say, for instance, delivering a plasmid in the cell that encodes for the ADAR proteins. And with that, we could start to then achieve pretty robust targeted RNA editing rates. But herein came the second challenge, which is that when we delivered just short antisense RNA, we could enable very precise on-target RNA editing. But when we delivered exogenous ADARs, this resulted in transcriptome-wide off-targets. Essentially, we had vandalized the transcriptome. And this is sort of, again, goes back to the fact that ADARs fundamentally only sense structure, and they're pretty much blind to sequence. And by overexpressing the ADARs, now these were being expressed at supraphysiological levels, and hence they were binding various other secondary structures, secondary RNA structures in the transcriptome. So to basically try and address this, Dhruva set out to then build a second generation of guide RNAs to recruit ADARs. And so the way he planned out these studies was, our goal was to basically try and achieve an RNA editing rate given by this dotted line. What this dotted line simply signified to us was that this was the kind of RNA editing rates that we would achieve if we gave exogenous ADARs into the cell, but we wanted to achieve this just using endogenous ADARs. So essentially the thought was, can we now design this new guide RNA that can help us reach here? So the first thing we just confirmed from our prior work that just delivering a long antisense guide RNA, you could get this about 10% RNA editing. Following this, we basically coupled various secondary structures that come from RNA that are known to be favorable substrates for ADARs, for instance, the ALU RNAs or GluR2 RNA. And each of these strategies of coupling additional secondary structures to these guide RNAs to sort of make them more favorable for ADAR recruitment enabled a slight modest increase in RNA editing, but didn't really achieve the kind of RNA editing that we wanted to achieve, which was given by this dotted red line. So at this point, what we conjectured was that maybe the recruitment of the ADARs was not the rate limiting step, but instead it was probably the stability of these guide RNAs themselves. And we were classically expressing these using polymerase III promoters, and the corresponding RNA, as we're all familiar, have very, very short half-lives. So at this point, Dhruva decided to circularize these RNAs. And by simply circularizing these guide RNAs, we started to then achieve very robust on-target RNA editing. And we were starting to now achieve RNA editing at the level that we would get if we delivered exogenous ADAR. So we were quite excited by this sort of observation. And essentially the way we engineered these circular guide RNAs was to use this sort of phenomenal technique developed by Liedtke and Jaffrey back in Nature, back in 2019. And essentially we use this strategy, which they call the tornado system, where we take our antisense region of interest, flank it with twister ribozymes, and these twister ribozymes then undergo cleavage inside the cell, exposing this 5' OH group and 2', 3' cyclic phosphate groups. These groups are then brought together by ligation sequences, and then this endogenous RTCB ligase can then trigger the covalent closure of this RNA molecule to give these circular RNAs. And one can confirm that these circular RNAs are generated inside the cell by basically utilizing these outward-facing primers And only in the presence of a circular RNA do you end up getting the desired band of interest. So as one would imagine with these circular RNAs, that perhaps these have increased durability. And so we indeed confirmed that these circular RNAs have this feature of increased durability inside the cell. And the second aspect that we confirmed also was the fact that this targeted RNA editing that we were obtaining was indeed due to the presence of the endogenous ADAR1. Specifically, we could deliver short antisense oligonucleotides to basically repress ADAR1, and correspondingly, the RNA editing rates also went away. So at this point, then we decided to check what was the kind of specificity of on-target editing that we were achieving using this methodology. And we were quite excited to see that using these circular guide RNAs, we had exquisite specificity for just the target RNA of interest. But what we also observed in our transcriptome-wide RNA-seq data was, while we had this on-target efficiency, very robust on-target editing as indicated by the blue circle, we also were observing off-targets. But interestingly, these off-targets were not on other transcripts, but they were actually on the target transcript itself. So what do I mean by that? So for instance, if I'm looking at my target RNA on the x-axis, and location 0 is my target site that I wish to modify from an A to a G, then when we deliver these circular guide RNAs, what you can see is at your target site, you get this desired conversion from an A to a G. But there are other flanking adenosines in the neighborhood of this adenosine that also get modified. And so we end up getting this bystander effect where all adenosines in the proximity also have a tendency to get converted from an A to a G. And this is really driven by the fact that you have this long antisense this long region of hybridization, and correspondingly, the entire region becomes a favorable substrate for the ADARs to dock and then modify all corresponding adenosines in that region. So this was a bit of a challenge because we only wanted this on-target editing. And while we had no other off-targets in the transcriptome, we had this massive set of off-targets at the target site of interest. So at this point, we were very inspired actually by nearly now a 25-year-old paper from the Brenda Bass lab, which was examining the role of ADARs and their ability to modify RNA in vitro settings. And so we were inspired by this study. And what we decided to do, motivated by this study, was to basically start to introduce short single-stranded regions by introducing these bulge structures on these guide RNAs in an asymmetric fashion. And so by introducing these bulge structures, one 5 base pairs upstream, another 30 base pairs downstream, essentially what you can see here, if you can see my pointer, is that all the off-targets in that vicinity essentially get eliminated. Essentially, these bulge structures are creating an unfavorable region for the ADARs to come and bind and modify RNA. And then one can subsequently just increase these, one can tag along additional, bulge structures and essentially just clear out all the off-target RNA editing. And in addition, one of the other advantages that we observed by using this sort of design was our on-target RNA editing rates also went up. So once Dhruva started to observe these results in vitro, our next thought was, can we extend these observations also into an in vivo setting? And in particular, what we did was we took these guide RNAs, we delivered them using adeno-associated viruses into mice to see if we could get in vivo RNA editing. So in the first set of experiments, we just targeted a test locus in the PCSK9 gene inside mice. And what was very exciting for us was that in a couple of weeks, we could already observe about 40% RNA editing, and this persisted for over 2 months of the study that we conducted. So we were getting between about 50% RNA editing on average at this target site of interest. So at this point, we switched focus to then think about, well, can we also achieve this in a mouse model of human disease? And in particular, we focused on this excellent mouse model of the type 1 Hurler syndrome, which has a nonsense mutation in the IDUA gene. And the very nice thing about this choice of this nonsense mutation in these animals is it actually coincides very well with also the frequent location at which these mutations are seen in humans as well. So there is this nonsense mutation, and essentially this nonsense mutation in the IDUA really results in a buildup of glycosaminoglycans in these animals. And this buildup of glycosaminoglycans is then associated with cytotoxicity and a whole host of other pathological effects. And so the goal with these studies then was to basically use these circular guide RNAs to try and reverse this nonsense mutation. And Dhruva was able to achieve about 10 to 15% targeted RNA editing in these animals with a concomitant decrease of the buildup of glycosaminoglycans as well. So as a brief summary of this portion of my talk, these circular guide RNAs can enable robust RNA editing across multiple sites, both in vitro and in vivo, across both untranslated and coding regions of RNAs. And these can vastly improve the efficiency and durability of RNA editing. And at the same time, one gets very high transcriptome-wide specificity because one is only recruiting endogenous ADARs. But then by using these asymmetric bulges on the guide RNAs, one can also enable a high degree of transcript level specificity as well. And these can be delivered both genetically encoded in DNA or even synthesized as antisense oligonucleotides and circularized in both in vitro or in situ. And we believe that these have a degree of therapeutic potential because these are very similar now to the ASO world or the RNAi world, where all the machinery for doing the RNA editing is actually now present inside the cell. We only need to deliver just these short RNA molecules inside the cell. And if we can effectively do that, then one can, in principle, enable therapeutic RNA editing. So this is an aspect that we are now focusing quite heavily on and trying to further improve this technology and move it forward. So switching gears now, I wanted to sort of talk about the fact that we are very interested in RNA editing, but we're also very interested in studying RNA editing in the context of development as well. And in this regard, I'm going to talk about the work of two very talented graduate students in the lab, Amir Dailemi and Sammy Liu. And essentially, what they wanted to look at was to think about, to basically study RNA editing in the context of early human development. And so the first thing that Amir and Sammy decided to do was to basically let's look at what is the RNA editing trajectories in the context of humans. And so essentially, we took bulk organs, looked at their RNA-seq data, and we just simply analyzed them for RNA editing inside the cells. And then one can basically observe these sort of trajectories where for some organs, such as, for instance, the forebrain and the hindbrain, there's a constant increase in RNA editing rates as one ages. But there are certain other organs, for instance, the heart, there's a decrease in RNA editing initially and then kind of remains flat. But there are certain organs, such as the kidney, where the RNA editing rates remain about unchanged throughout the course of the individual. And of course, this was done primarily using tissues and publicly available data sets. But our thought was, can we actually now start to look at this more closely in the context of studying early human development? And in this regard, one of the big challenges that one faces is the fact that if you want to study the impact of certain genes in development, one really needs to be able to traverse sort of three trajectories, or rather three major aspects. The first is we want to be able to introduce these perturbations. But then we also want to be able to collect a large number of phenotypes. And a third major challenge is to also be able to do this in the context of many different cell types. And this is where model organisms can be fantastic because one can introduce these perturbations, one can look at the whole gamut of very complex phenotypes in these model organisms. And importantly, one can do this across the whole context of the organism. And this, of course, is a challenge for human development. And so recently, a few years back, we had proposed that perhaps one could use teratomas, which are basically complex tumors that are derived when you take pluripotent stem cells and you inject them inside mice. And then these complex tumors basically have the whole spectrum of cell types from all germ layers present in them. For instance, if you do a single cell RNA-seq, you see this whole spectrum of cell types, over 25 different cell types across the mesoderm, endoderm, and the ectoderm. And so these are also a very easy model to implement in the context of multi-lineage human development. So we basically then decided to use these models of development to try and look at the role of RNA editing. And so we did two aspects. The first was we took human embryonic stem cells and we generated cerebral organoids. And in parallel, we also generated teratomas. And you can see when you look at, say, a 10-month-old cerebral organoid, you can see the various, the four major sort of cell types that are classically seen at this time point. And you can start to look at the different degrees of RNA editing in these various cell types. For instance, in excitatory neurons, there's a high degree of RNA editing, but in inhibitory neurons, this is much lower, primarily driven, at least in part, for instance, by ADAR3 expression. But of course, here we looked at cerebral organoids, but if you now look in parallel at just simply 10-week-old teratomas, one can now see that instead of just the four cell types that one sees in the classic cerebral organoids, one can now see all of these additional cell types as well in the teratomas. And additionally, and very excitingly, one can start to recapitulate certain observations that have been seen in the context of human tissues, where if you look at the muscle, there's much lower RNA editing rates inside muscle. One can also then see that, of course, as I indicated, muscle has much lower RNA editing. and it is lowly correlated with ADAR expression. But in fibroblasts, for instance, which don't have ADAR3, an inhibitor of RNA editing, this is much better correlated. But now if you move into neural progenitors, this correlation again goes away, primarily also driven by the fact that there's ADAR3 expression here. And we also confirmed that both the single cell RNA editing levels that we were observing were very concordant with what one would observe if we just did bulk RNA-seq and looked at the corresponding RNA editing levels as well. So next, we basically generated teratomas over a time course, looking at week 4, week 6, week 8, and week 10 teratomas. And then we looked at RNA editing over time. Across these various time points, one can essentially see that more mature cell types come up and more early cell types start to go down. And correspondingly, if you look at the RNA editing levels, one sees a gradual increase in the degree of RNA editing over time. But of course, one advantage that comes by using these models is one can actually go in and start to do perturbations. And so in this context, we basically did a CRISPR pool library targeting both ADAR1, ADAR2, and ADAR3 in these teratomas. And then we looked at these teratomas at the end of 10 weeks for looking at the impact of these ADAR knockouts, both on cell fitness or cell fate specification, as well as the impact on transcriptomes as well. And so the first, just looking at RNA editing, you can very quickly appreciate that by doing an ADAR1 knockdown, the global RNA editing rates in these cells basically go down. But interestingly, now one can start to look at the impact of ADARs on direct cell fate specification. So just to sort of conclude by one vignette here, one can actually start to observe, for instance, that when one does ADAR1 knockouts, one has a predominantly greater effect in the mesoderm as compared to the ectoderm and the endoderm. And this was quite interesting to us. And for instance, you can actually start to see that ADAR1 has a big impact on the fitness of the cells in the mesoderm as compared to any of the other ADAR2 or ADAR3 knockouts. So this is, of course, an ongoing body of work, and we're still trying to parse through and see what all of this data means. So I just want to quickly conclude by stating that we have utilized here multilineage human pluripotent stem cell differentiation to characterize RNA editing at single cell resolution and over time during early human development. And by directly sort of perturbing these ADAR family members in multilineage models of differentiation, such as those via teratomas, using pooled CRISPR-Cas9 screens, we're now starting to gain insights into their role in both cell fate specification as well as RNA editing in this early developmental time window. So with that, I just wanted to acknowledge my research group. I've already highlighted the key folks along the way. Our close collaborators, the Kun Zhang Lab and the Michael Green Lab, our funding sources, my mandatory disclosures, and I'm happy to take questions if time permits. Thank you.
Thank you, Dr. Mani. This is a wonderful talk.
Thank you.
I didn't see any question in the Q&A section. I actually wondering, can you, when you look at those A2I editing, did you see some nano modification happen at the same time? Have you guys looked into that?
Yeah, that's a great question. So actually, we were only looking at A2I editing. So we basically just do cDNA conversion. So at that point, immediately we're constrained with the kind of modifications that we can detect. So we've only looked at A2I changes. We've not looked at any other modifications that are getting impacted at that target site.
Okay.
Sheng-Long, I asked a question in the wrong format. I put it in the meeting chat, and my question. This is Scott Blanchard. I was asking. This was a brilliant lecture. I really enjoyed it. How can one most naively assess whether ADAR activities are at play in a given biological setting? Is there some telltale signature that you would look for, either in proteomics or RNA-seq, et cetera?
Yeah. So actually, here, so one index that, and I'm sorry I didn't dive into the details there, is we looked at this index called the ALU editing index that actually was developed by Erez Lavenon and colleagues. It's a wonderful way to just look at or have a proxy for the global RNA editing rates inside a cell. And so we really use that as a metric to look at the degree of RNA editing in a cell. So if maybe that answers your question to a degree, I think, where, and there, we can actually show that, if you do an ADAR knockdown, for instance, then the corresponding ALU editing index goes down as well. So we're currently using that as a nice proxy for—
That's really great.
For measuring RNA editing, but this is really work from Erez Lebanon and colleagues who basically really developed that technique.
Can you say the name again?
I
We
can
have
type
to
it in.
move
I can
on.
type it in. Yeah, I can type it in. Yeah.
Okay, that's great. Thank you.
Once again, thank you, Dr. Moni, for this wonderful talk. The next speaker would be Dr. Yitao Yu from University of Rochester Medical Center. And he will talk about pseudo-uridine modulated nonsense suppression. Whenever you're ready, you can take away.
Oh, okay. Now I think, can you hear me?
We can hear you.
Okay, let me see if I can get a
So that's
screen.
in the studio.
Okay, 1 second. Now, can you see the screen?
Absolutely, yes.
Good.
Okay, good. All right. So, thank you for the introduction, and also I want to thank Sammy and Kate also for inviting me to give a talk about our work here. The title of my talk today is I changed this a couple of times now, finalized this but it's not perfect anyway. So it's modulating gene expression by targeted RNA citylation. So, doesn't go. Oh, here. Let me just get the laser. Okay. So, just wanted to go quickly because the pseudo-U has been introduced in the previous talks, but I wanted to go quickly here. So pseudorylation is an isomerization reaction that converts uridine into pseudouridine. In pseudouridine, you have like a extra hydrogen bond donor here and a carbon-carbon linkage here. So this reaction endows the modified nucleotide pseudouridine with the chemical properties that are distinct from that of uridine and of other nucleotides. So as you can imagine, if it's incorporated into RNA, it can alter RNA function. So pseudouridine is identified, has been detected in many types of RNAs, including messenger RNAs, coding RNAs, messenger RNAs, and the non-coding RNAs. But they are mostly concentrated in ribosomal RNAs and snRNAs that actually required for pre-messenger RNA splicing. For example, U2, vertebrate U2 RNA, in this region, which is a branch site recognition region, during splicing, it recognized the branch site sequence of a pre-messenger RNA. In this region, all six uridines are converted to pseudouridines. I'm going to come back to this later. So a pseudorylation is catalyzed in these RNAs. It's catalyzed by a mechanism we call this a RNA-dependent mechanism, or it is a guided mechanism. The guided RNA is the BOX-HACA RNA. There is a family of these RNAs, although they have the different sequences, but they can all fold into this structure called hairpin hinge, hairpin tail structure. And each of those RNAs can be complex. Each of these RNAs is complex with 4 core proteins to form RNP. The proteins are NHP2, NOP10, GAR1, and CBF5. CBF5 is actually the enzyme that catalyzes the chemical reactions during the conversion U to pseudo-U change. So during pseudorylation, the internal loop sequence of the guide RNA, or we call it the pocket, pseudorylation pocket, that base pairs with the substrate RNA, specifically positioning the target uridine at the base of the upstem, that tells the enzyme CBF5 which uridine to be converted to pseudouridine. So this is a highly specific reaction, and the specificity is determined by the base pair interaction between the guide sequence and the substrate sequence. So in principle, in theory, And you can take any of those naturally occurring guide RNA and change the guide sequence only to redirect the modification to different sites or even different types of RNAs. So today I'm going to be talking about how we use this technique to study or to test whether pseudo-U plays a role in gene expression. I'm going to talk about two projects currently being carried out in my lab. One is about snRNA pseudorelation and pre-messenger RNA splicing, and the other is nonsense codon pseudorelation and nonsense suppression. Now it goes to the first. As I also, I mentioned earlier, this is a vertebrate U2 RNA, and in this branch site recognition region, all 6 uridines are converted to pseudouridines. But interestingly, in yeast U2 RNA, only 3 of them are converted to pseudouridines. So why there is a difference here? So now if we bring the substrate into this picture, now probably it makes some sense. And here again, this is a branch site recognition region U2, and the pair pairs with the branch site sequence in pre-messenger RNA here. As you can see, the branch site sequence in pre-messenger RNA is very loosely defined, or it's very degenerate. So it's actually there are a lot of different sequences that can be used as a branch site sequence. So how come then you have one sequence can recognize many different branch site sequence? So, but now if you think about it, because pseudo-U is actually very flexible in pairing with the nucleotides, and it's being reported that in addition to A and a G, pseudo-U can also pair with the uridine. So it probably brings more flexibility to pair with the different sequences here. On the other hand, we also detected that we determined that at any given site, pseudorelation is not 100%. It's about only 80 and 90%. So, and in combination, you can imagine that there's a lot of different sequences even in U2. Although you think there's only one sequence, but if you bring this pseudo-U into the picture, then there are a lot of different sequences, and this is probably why I can recognize many different diversified branch site sequence here. So in yeast, you have 3 pseudouridines in U2, but now you look at the branch site sequence in pre-messenger RNA. There is only one sequence. There's very few alternative sequence served as a branch site sequence here. So that makes sense, right? So we decided to test this by doing a spring experiment. So we decided to do this in yeast cells because it's easy to manipulate the pseudouridine in yeast U2 RNA. So we can either, because of 3 pseudo-U enzymes are known, then we can delete them then to block pseudorelation at these 3 sites, or we can introduce the guide RNA targeting these normally not pseudorelated sites to make them pseudorelated, so that we can even make it like all 6 pseudo-U's here, mimicking the mammalian U2 RNA. So this is the idea to introduce the guide RNA to target those normally unpseudorelated sites and to introduce pseudo-U's. So this is the example showing you that this is the CMC modification followed by prime extension analysis to detect the pseudouridines in U2 RNA. Normally you have the 3 pseudo-U's in yeast U2 RNA in the wild type. Now, if you knock out one enzyme, then you lose this modification at this site 42. Now, if you knock out another pseudo-U enzyme, now you lose a modification at this site, 44. Now, if you knock out both, then you lose modification both sites. Now, on top of this, if you introduce the guide RNA targeting these 2 sites, now you can restore the pseudorelation at these 2 sites. So now on this panel here, this is under the wild type background. You already have the 3 pseudo-U's. Now you introduce 2 guide RNAs targeting the 3 unmodified sites. Now you detect all 6 pseudo-U's there. So we can easily manipulate the pseudouridines in these DU2 RNAs. So to do screening, then we actually decide to take advantage of the strain called the CUP1 deletion strain, because CUP1 is a chelator that can chelate in the copper and allow cells to grow on the CUP-containing medium. So if you delete this CUP gene, then the cell will die in CUP-containing medium. It becomes copper sensitive. So now, if you introduce a plasmid which contains a fusion gene, actin-1 is fused with copper-1 and with the intron brought by actin-1. Now, this plasmid, once it's transformed into cells, then it will be transcribed into pre-messenger RNA, then it will be spliced, and then produce the fusion protein. Because the fusion protein is functional, then it can reverse the copper sensitive into copper resistant, depending on the amount of the protein, and it can allow cell to grow under different concentration of the copper medium. So, and also the copper protein production is also pretty much correlated with the efficiency of splicing. So the efficiency of splicing can easily be read by the copper sensitivity, right? So, and here, because splicing is catalyzed by spliceosome, with those spliceosome SNRNAs involved, and all we did here is just changing the pseudo-U content in U2 RNA. So if this change or different combinations of different number of pseudo-U's introducing U2, if that affects the splicing, it should be reflected in the copper sensitivity. Right, so here's the strategy, and this is what we did. And this is a pre-messenger RNA, and then we randomize this sequence and shown here. Then we transform it into the CUP deletion strain. And of course, this is just one strain, and we have many different strains with different U2 background. And then we can plate and doing a replica plating and into different CUP concentration medium, and then collect the cells and then isolate the plasmid sequencing. Then we determine and we can select the sequence of the branch site sequences. And this is just one example that we did using, this is under this background, this strain, U2 with no pseudo-U, exactly as expected. And when you have increased the copper concentration, you have fewer and fewer colonies. So, okay, so this way we generated a large amount of data and get a lot of the different branch site sequences and shown here. This is a heat map here. So at least, by looking at this, we can see, I wanted to point at 2 points. And the first is that for most of those sequences and can be clearly differentially recognized by different U2, and different U2, I mean the different U2 pseudo-U content, right, in U2 branch recognition sequence. So, and the other point is that the order of favorability of these sequences is different in different strain and the different U2 background. I'm saying this by saying this, for instance, these two sequences, right? It is more favorable in this strain, and this sequence is more favorable in this strain over this sequence. But in this strain and this background, and this is reverse, right? This sequence now is more favorable than this sequence. So this really suggests that pseudo-U plays a regulatory role in selecting the branch site sequences. So we actually tested a couple of those and see that exactly we put those branch site sequence side by side and test them in different strains, and we found that indeed under one strain that one branch site sequence is favored, and on the other strain that the other one is favored. So we're still digesting the data, and hopefully we can make more predictions to test. So now I'm going to switch gear to the second project. And in the first one, we just like using the GARD RNA to target the different site in U2 RNA. But now we're targeting the different types of RNA, and we did many such experiments, and we're particularly interested in the targeting messenger RNA because messenger RNA carries genetic information, and we're thinking that if we target targeting or incorporation of the pseudo-U into messenger RNA into codon, and probably we're changing the coding specificity. We first, of course, focus on this stop codons because all three stop codons have the uridine at its first position here. So for targeting. So here shows the idea here. This is DNA, and once it's transcribed and processed into mature RNA, it will be translated into protein, and it will stop here because it's stop codon, right? And now we are targeting this uridine based on the sequence, right? Design a guide RNA targeting this uridine, and to convert this into pseudo-U, and then we're asking whether this is going to change the coding specificity. If it does, then ribosome will go through because it becomes a sense codon. So we'll detect and extend the version of a protein, right? So to test this, we did many experiments in yeast first, using several different systems. And this is one example we did several years ago. And in this experiment, we're using the TRIM4 reporter system. And the TRIM4 gene actually was derived from, obtained from a Great Hack in the physics lab in a neighboring lab. And this gene was fused with the TAP-TAG here. So you generate the TAP-TAG, the fusion protein here. But now we introduce the PTC, premature termination codon, either one of these, into the TRIM4 gene at the C-terminal end. And then translation will stop here, and no fusion, tag fusion protein produced. Now, we introduce a guide RNA targeting this site, the stop codon here, PTC. And then once it's in the cell, it will assemble into a particle and then convert the uridine into pseudo-U. And if this is sufficient to allow stop codon to change back to sense codon, then we will expect that the fusion protein will be produced again. So the tag will be used as to measure the read-through, the ribosome read-through. So here is the result, and this is just without the wild type, without the premature termination codon. You see, it's very efficient to produce this protein. This protein is completely no full-length protein produced, no fusion protein produced when we introduce the premature termination codon there. However, if we introduce the guide RNA targeting this premature termination codon, now we detect the read-through protein. And this level is relatively low. And we did this many years ago. And now by triggering here and there the guide RNA, we can get the efficiency much higher than that. Okay, so all these experiments were done in yeast, and then we're asking if there is any clinical applications. There is a large number of genetic diseases caused by mutations, and 15 to 20% of those mutations are usually are actually nonsense mutations, and the list of those disease genes are very long. And here's the illustration here. So this is double-stranded DNA again. This open reading frame, start codon, stop codon. And the first, it will be transcribed and processed into mature messenger RNA, and it will be translated into protein according to this open reading frame. Now, in patients, and some of those nucleotides, well, one nucleotide, let's say, has a point mutation here, and this C changes to T, say, then this creates a nonsense mutation, right? Because this is a nonsense codon. At the RNA level, it's a UGA, it's a nonsense codon. So that will trigger the surveillance pathway called nonsense-mediated mRNA decay that degrades this nonsense-containing messenger RNA. And there's a small fraction of messenger RNA will escape this degradation, and they will be translated because this is a stop codon, translation will stop right here. So in patients suffering from the nonsense mutations, And though they're essentially no full-length protein in patients. So this is most likely the very severe type of disease. So what we are asking is that if we change this uridine into pseudouridine based on the sequence, we can design the guide again, right? Change this into pseudo-U. If this is sufficient to convert this nonsense codon back into sense codon, and then we will believe that we'll block the NMD because it's no longer stop codon. And then we'll accumulate a messenger RNA. And then because it's not a stop codon, then translation will go through and until it hits the normal stop codon. And that will restore the full length protein production. So to test this, and we did this experiment using the beta thalassemia gene and derived from Lima Quad's lab, our neighboring lab again. So this gene, and this is a pre-messenger RNA, and this is the premature termination codon here. And this is the mature messenger RNA, and this is the sequence surround. This is actually the premature termination codon, UAG, because resulting from nonsense mutation. And this is surrounding sequences. Based on the sequence, then we can design a guide RNA targeting this uridine, right? So let's take a look at the results. Okay, here, this is the wild type beta globin gene, and without this premature termination codon. As you can see, normally you have this level of messenger RNA detected and this level of protein detected. But now if we introduce this nonsense codon here, now virtually very little amount of messenger RNA detected. This is not unexpected because it is degraded by NMD. And virtually no full-length protein is detected. But now if you introduce a guide RNA specifically targeting this uridine, the nonsense codon, now you see the messenger RNA level is brought back to some extent, and the full-length protein is produced. So we test this in many different disease genes and nonsense codon suppression, right, in disease genes, and also in many different model cell lines. And we got similar or consistent results suggesting that the nonsense suppression occurs in the other genes, the other disease genes in model cells as well. And now,
sorry, Doctor, you have to wrap that up.
Yeah, I don't have time to talk in detail, but just like conclude. This is the summary. Just want to bring two major points here. One is like both messenger RNA and the non-coding RNAs can be site-specifically pseudorylated by designer box HAC RNP. And the second point I want to make is that the pseudouridine plays an important role in modulating gene expression. I gave you two examples already. One is the S9A modification and a pre-messenger RNA splicing, and the other is nonsense codon modification and nonsense suppression. And finally, there's acknowledgement. And so I want to acknowledge the people current and the former, and these people are involved in the projects I talked about today. But thank you.
Yeah, that's a wonderful talk, Dr. Yue. Maybe in terms of time, I see one question. Yeah, seems like there are many different ways to sequence pseudouridine. I'm wondering, can you talk a little bit why you're choosing the method you are currently used for sequence the pseudouridine you're working?
Oh, you mean the pseudo-U method that we used is CMC modification by prime extension, right? So that mapping pseudouridine. I mean, this is very efficient assay we always use because we know where pseudo-U's are, and they are in what region at least. Then whatever we detected, then we focus on that region, and it works for us really well. And of course, you can do many different ways of the sequencing, but of course, we don't need like a high throughput at this point to detect the pseudouridine U2 RNA, for instance. But well, for messenger RNA pseudorylation, and because we target the stop codon, right, we also did the high throughput sequencing. And it's also, yeah, I mean, this is just also based on CMC modification and prime extension. And of course, you can use a different, assay to detect it. And then the point is that we use that assay, we detect the CDU at the expected site, and we didn't detect any significant off-target problem effect.
Interesting. Thank you, Dr. Yu, for sharing this. So in terms of time, we have to move on to the last speaker. But I want to remind, I think Sami and Kate, they're going to make a brief closing mark at the end of the session. So stay tuned. So next speaker, the final speaker of the symposium would be Dr. Jiaxi Huang from Duke University, and he's going to talk about a programmable RNA sensing for cell monitoring and control. Whenever you're ready, take away. Dr. Huang.
Okay, hope you can hear me and see the slides. First, I would like to thank Sami and Kate for inviting me to speak at this last session, which is quite appropriate for my talk. So I'm not an RNA biologist, or at least not yet. I hope I'll become one soon. I'm a neuroscientist interested in studying brain circuits by dissecting the diversity of cell types. And so we develop technologies that leverage RNA profiles to monitor and control cells, but this is of general interest and a challenge in biology and medicine, hopefully of interest to this audience. So today I would like to present a technology that we invented recently called Cell Reader. And in a nutshell, it's a programmable RNA sensing method that is coupled to effector protein translation. And the goal is to then monitor and manipulate the cell according to the RNA profile. And we hope that this will allow us to read and write cells in tissues in a way similar for gene editing to read and write genes. So again, the focus here is on the cells rather than the genes, either the DNA or the RNA. And the background is, as we know, that the advance in single-cell RNA sequencing and others promise to review the RNA expression of all cell types in the human body and in many species, and also in disease states. And the opportunity and the challenge is, what can we do about this amazing data set in biology to discover cell type function in many contexts, and in medicine to achieve precision therapeutics. So what we need is our technologies, just like CRISPR in gene editing, that can apply to cell types and cell states that are specific, easy to use, scalable, and general across species, including humans, and importantly, programmable. So for many years, my lab has been developing genetic tools to study brain cell types. And the starting point is by picking a marker RNA in a cell, and then typically go to the genome of regulatory elements to generate transgenic lines or gene targeting knocking lines or enhancer elements. And there have been advances, but one of the fundamental limitations is that this is fundamentally an indirect approach to recapitulate RNA expression through genomic DNA, and it's, we all know, quite cumbersome and can only be applied to limited species. So we have been thinking for some time whether we can develop RNA sensors that can detect the presence of cell-defining RNA directly, and then couple this to the translation of effector protein, essentially turning a DNA transcription-based approach to an RNA translation-based approach. And briefly, this is achieved by developing a new RNA sensing technology based on RNA editing. So it is a single RNA molecule with two domains. One is the the 5' is a RNA sensor domain. Oops. That is complementary to a cell-defining RNA, and the 3' is a effector translation domain, and this is separated by a self-cleavage coding sequence T2A. And the key feature is that there is a translation stop codon in the middle of the sensor sequence. So normally this RNA will be inert when it's delivered into the cell, but in the presence of a cell-defining RNA target, this will form a stretch of a double-stranded RNA, and as we have heard from Prashant Mali, that this is a substrate to recruit the RNA editing enzyme that's already in the cell. And specifically, we design A to C mismatch at the stop codon, which is a particularly favorable substrate for ADAR, which then convert adenosine to inosine, converting a stop codon to a tryptophan codon. And this allows the translation of the 3' effector protein. So essentially is a target RNA dependent trigger for translating desired protein. And the advantage is that it's delivered as a single RNA molecule, as ADAR is already in the cell, and also it is entirely sequence-based. So it has a number of advantages. One is that it can be very specific to the RNA and to the RNA defining, to the cells defined by an RNA, and it's easy to design and build. And it can be scalable for many RNAs, many cell types, and also across species, as ADAR is a ubiquitous RNA editing in all animal cells. And also it's programmable in the sense that once a sensor is defined, it can be linked to different effectors to manipulate or monitor the cell in different ways. And also one can envision building a sensor that needs to detect two targets to express the effector. So this can increase the specificity of cell definition. And also one can deliver a cocktail of these RNA machines to target multiple cell types, but instruct each cell type to do a different but concordant thing to modulate tissue function, for example. And so this first needs to be delivered inside the cell. So currently there are two main strategies. One is to deliver from a DNA expression vector. So far we have done all with AAV. And the other is using lipid nanoparticles, others to deliver the RNA directly. So the current deliver vehicles may have certain surface properties, but typically those can only achieve a so-called regional specificity, where the ultimate cell type, cell state specificity can be realized by RNA sensors. Why is it not? Okay, yeah. So this is the proof of principle and this is spearheaded by a very creative postdoc, Yongjun Chen. So he first implemented this method by in HEC293 cells by using exogenous RNA targets. This is TD tomato, and then the sensor will include a BFP expression vector to label all the transfected cells, then a sensor, which is complementary to the TD tomato sequence with a editable stop codon, editable A here in the stop codon, and then the readout effector is a GFP protein. So as you can see, only in the double transfected cells, there's GFP translation, and this can be assayed quantitatively by FACS. And indeed, it is due to a conversion of A to I that removes the stop codon. And he then showed that by changing the concentration level of the BFP using an inducible system, this can increase the rate of editing. And the length of the sensor needs to be about 200 to 300 nucleotide. So it is a rather long sensor, much longer than the small RNAs, the CRISPR guide RNA, and it can tolerate about 5% of mismatch, which give us some flexibility for the sensor design. And also, there are potential leaky translation in the sensors, so we can build in 2 or 3 stop codon to reduce the leaky translation. Skip this one. So he showed that not only this works for exogenous RNA targets, but also endogenous RNA targets in HEK cells. In this case, he built sensors across the transcript from exons to introns to cDNAs to UTRs. And they all work to some extent. And we are actually exploring this further, as I'll mention towards the end of the talk. And then these sensors can detect both highly expressed RNAs in HEK cells, such as this EF1A, also moderate or even lowly expressed transcripts. And also, interestingly, we can design an AND gate where the sensor needs to see 2 endogenous targets. One is EF1A1, the other is actin T-beta. So 1 is not enough, but only when 2 are there that the sensor will integrate and translate the effector protein. So of course, the key question is whether they can work in vivo. And here we leverage our expertise in neuroscience. My lab studies the various cell types in the mouse cerebral cortex. We developed a sensor that target a transcription factor called CTIP2, which labels the major types of layer 5B cortical output neurons that send projections to spinal cord, pons, thalamus, and others. And here we use a binary system. So the sensor first make a effector, which is a transcription activator. And then this signal can be amplified by co-injecting another AAV that express either GFP or other effectors from the TTA response element. So here you can see when we do a double infection, these are the transfected neurons that express the marker mCherry. And then a subset of these cells express GFP. And then we use a CTIP2 antibody to assay the specificity. As you can see, these expressed in the deep layers. And the specificity in this case reached over 90%, where most of these GFP cells are CFP positive. And in this local region, over 80% of the CTIP2 neurons are infected correctly by this GFP. And to show that this actually can be used to monitor and manipulate the cells, we express a genetically encoded calcium sensor called GCAM in this somatosensory region of the mouse cortex. And then we show using fiber photometry imaging outside of the skull that by stimulating the mouse forepaw, we can induce and record a time-locked calcium signal from the cortical surface. And finally, furthermore, we can control these cells. We can introduce a channel rhodopsin into these cells using this cell reader system. And then, as you can see, we can shine light outside the skull and to induce the falling movement because this area is known to control the falling movement. And then another question is whether this can be generalized to other species. So we have shown that this works in rat cortical neurons and also more recently in macaque monkey. But in particular, in collaboration with neurosurgeon Derek Southwell in our department, we show that it can work in human cortex ex vivo tissue. So Derek is he treats epilepsy patients. In many cases, he had to resect the epilepsy foci. This brain tissue typically is discarded, but in fact, it's a very valuable tissue for studying the human cortex. But previously, using general methods, they can only target neurons broadly, which is a dense forest where the diversity of cell types allow them do not allow them to study a neural circuit organization. So here we have developed 2 sensors, and now we have developed a dozen sensors. One target this FOXP2 transcription factor, which labels a set of layer 6 cortical pyramidal neurons, and also a superficial layer glutamatergic neurons that allow them to record and actually channel using optogenetics to manipulate those cells. And also we develop another sensor targeting this vesicular GABA transporter, which is expressed in all the GABAergic inhibitor neurons. As you see here, these cells have a completely different morphology and also firing patterns. So in that sense, this technology allow potentially us to read cell states almost like the RNA barcode using like a QR code detector, and then to write a cell property accordingly. If this is a cancer cell, we may want to kill it or label it for immune cells to kill it. And because this modularity, it allows us to have a platform potential to detect many cell states and disease states, and then according to that state to act accordingly by expressing the appropriate payload. So currently we have identified sensors for over 70 genes, and this is still growing, testing over 500 sensors in 5 species and multiple tissue types, including mammals, birds, and more recently, reptiles. So for this audience, I want to say that this is really the beginning of a new technology. There are many areas that needs to be improved. Like many technologies, first and foremost, specificity, efficiency, and efficacy. And so we need to establish design principles and algorithms. And first to define the rules and the constraints of interactions between sensors and targets. And these are large RNAs rather than small guide RNAs. So it is a new territory across the RNA life cycle. And we hope to do this by implementing sequence-based Hesper assays to screen large RNA libraries and to target many RNAs in cell lines and also in tissues. And then using a computational or machine learning approach, hopefully to design, to derive algorithms. And the overall idea is to achieve a holistic design that consider both the primary sequence and perhaps the high order architecture of the RNA, especially the target RNA, to achieve specificity in RNA sensing, efficiency in this editing, and efficacy in the translation. And the overall goal is to achieve easy, reliable, and scalable design that can be applied across cell types and species. So as many of you are expert here, when you think about this, it's actually surprising that it works and also seem to work quite reliably, because transcription is in the nucleus, and then there's RNA capping, editing, modification as the topic of this meeting, splicing, and then export translation. And through this life cycle, this RNA is covered by many RNA binding proteins. So really, where and when can RNA sensing occur? So we decided first to probe this by designing sensors across the target RNA and to assay whether it can occur in the introns, in the nucleus, in the supreme UTR, axon, and all that. And so literally by picking transcripts that have a characteristic length, intron axon structure, supreme UTR, and we're in the process of trying to test this, and then to achieve a larger scale screening using sequencing-based methods. And the other is really to achieve biological effect, either monitoring or controlling or therapeutics effect. We need to make enough of the effector proteins. So many aspects of the cell-ready RNA need to be optimized from 5' UTR to 3' UTR to others, and many people know here. And potentially use of circular RNA, as we also have heard from Prashant. And also there's an interesting design recently from Jim Collins' lab, where they put a additional ADAR sequence as part of the payload. And so after sensing the target, this part of this payload will come back to amplify the sensing and effective translation. And I mentioned this not because we have already done this, but hopefully really to get feedback, interest from this audience, either here or after the meeting, write to me. And I just want to acknowledge that this has been spearheaded by a very creative postdoc, Yunjun Chen, who now just started his own lab in Peking University Center for Future Technology. And I'm very interested in engaging the students or postdocs here who are interested in this work. Please write to me. And these are my funding. So I'll stop here.
That's a wonderful talk. Thank you, Dr. Huang. Yeah, I may start with one question, hopefully to see, give time for people to write their question in the Q&A. I have a question about the mismatches in the guide RNA. Can you talk a little bit about how the mismatch in the guide RNA impact the specificity, efficiency? Can you talk a little bit about this?
Yeah, that's an interesting question. So far, we have not really rigorously compared the sensors with or without mismatch. We only know that with a few mismatch, we can still get RNA sensing, certainly in the cell and in other cases in vivo. And we mainly take that as some flexibilities that allow us to design sensors in some flexible ways.
Great, thank you. So if there's no more question, I would like to thank the great speaker, Dr. Mali, Dr. Yu, and Dr. Huang. So we'll conclude for this session 3. I will yield back to Sammy and Kate.
Thank you, Dr. Zhang. I appreciate it. Sure, just one moment and I will set up Dr. Jaffrey and Dr. Mayer.
All right, wonderful. I just want to thank everybody as we're getting set up. You know, everybody's rushing to close their window, but I just want to thank Eileen for organizing and the AV department for organizing seamlessly this conference. And I know I'm going to appear in 1 second, but I want to thank all that hard work. I want to thank all the speakers. And I just want to let you know that, you know, hopefully we'll see you again in probably 2 years where we do another methods-based epitranscriptomics conference. And Kate, and I also want to mention we had about 700 unique registrants. It came in at different times, but it was really well attended. Kate, do you want to say anything?
No, I think you said most of it. Just thanks to everyone. Thanks to Eileen and the AV department who really helped coordinate everything. Thanks to all the speakers. And a few of you have asked me about the recording. Once we get permission from all the speakers, we will send out information on making the recording available and how you can access it. So stay tuned.